everybody, this is our last video for chapter 2. We're going to talk a little bit more about research methods and some issues with research, and we're going to talk a little bit about ethics. So our first slide, once again, we're going to revisit this topic that not all questions about human behavior can be answered by the scientific method. For example, abortion, should it be legal? Will I go to heaven when I die? Is it important to study psychology? Answer to that one's yes. Okay. These are values. These are value statements. They are not facts. So values are not things that can be objectively measured. So these are not things that are found through scientific research. And we did talk about this in another video. So it's really important to be able to identify the difference between a value statement and a factual statement. So that's just something I've kind of tried to bring to the surface throughout our videos, because that's something you need to keep in mind when you hear people say something. Once again, most of us present our values as though they are facts. And that's just kind of a human nature thing to do. But remember, facts are objective. They can be determined to be true through empirical study, and they are basically something that could be falsifiable. You know, they're something that can be proven wrong or supported to be correct. Using the scientific method is how you determine whether something is an actual fact. Okay. And we'll go to our next one. There we go. I couldn't get it to click. I want to talk a little bit more about avoiding biases. Experimental bias is when researchers' expectations skew the results of the study. There's participant bias, which is when a participant's expectations skew the result of the study. There's something called a single blind study, where the researcher knows which participants are in the experimental group, which are in the control group, and what exactly is being studied. And then there's a double blind study where the participant in the experiment and the researcher that is conducting it at that time doesn't know who is in what group. The reason it's important to do a single blind or a double blind study is because we can accidentally affect the results of a study or accidentally influence a person's behavior unintentionally. For example, especially if you're working with like kids, when you're working with kids, we're going to look at an experiment later that shows a child having to choose between two stuffed animals. They had watched a little video. One of the stuffed animals behaved in one way, one behaved in another way. So one of them was a helper animal, one of them was not so much a helper animal. And then at the end, the small children were supposed to choose which one they liked. So the researcher would present the two stuffed animals on this tray, and then the child will select which one they liked better by grabbing it. If an experimenter knew which group the child was in, or if the experimenter knew which one of those animals was the good guy, which one was the bad guy, they may inadvertently influence that child with their selection of that stuffed animal by maybe making a face or tensing up, or maybe tilting the tray slightly towards one animal or another, so that it's closer to the child. That is not something that people would do on purpose. Usually that's something that we would do unintentionally. Our body language says an awful lot to other people. We don't just communicate through voice and words. Our body language and our facial expressions are huge with communication. So experimenter bias can be controlled by that double blind study by making sure that the person that's like presenting those animals doesn't know like which one of those animals is the good guy, which is the bad guy, because that makes sure that they don't accidentally influence the outcome. Participant bias is something that we also have to be very careful of. In psychology, often we use a little bit of deception. In theory, we should try to avoid using deception anytime we can. However, when we're talking about human behavior, when people know what you're studying, oftentimes it will influence how they behave during that experiment. We're going to take a look at another study later on in our course where people are told that they are going to be looking at, they're going to be doing a study that talks about like sense and perception. And they're supposed to be looking at some different lines, and they're going to match the length of one line to another line that they were shown. So they're given this one line, they say, which of these lines matches that one? So people go in there, they think they're being tested on their perception, like how they perceive those lines and being able to select the correct one, when actually it's going to be a social experiment. And the other people in the room are actually with the experimenters, and they're going to behave a certain way to see if they can influence that person's choice. So if the person knew that that was happening, the research would not turn out right, because they would end up having some sort of bias, or they would be not changing their choices because they would know that we're studying whether or not somebody could change their choice. So anyway, so that would be an example of participant bias, and we will take a good look at that later. So those are things that you just have to be careful about when you are conducting research. There is something called the placebo effect, and I'm going to give you an example of that. A placebo effect is when you guys have all probably heard about drug trials, where one group is given a particular drug, the other person is given what they call a sugar pill. And in that case, that's how you test whether or not a drug is working. So I've kind of put together a little example on the screen here. So in this particular study, we're going to talk about a drug study and what the placebo effect is. Our independent variable is the drug type. So remember, our independent variable is the thing that is manipulated. It is something considered to be the cause in our cause and effect relationship. So our drug type is going to have two different quantities that we give. So, or in this case, we're going to give our experimental group the 20 milligram per kilogram of the drug, and our control group is going to get a 20 milligram per kilogram of saline, if we were doing injections, or maybe just a same like 20 milligrams of an inert substance in a pill form, so that there's no actual drug in that. They're getting the same quantity, but it's not a drug. And they're getting administered in the same way. So if people were getting injections in this study, you would either get an injection of the drug or an injection of saline that does not have any drug in it. So remember, our experimental group and our control group. Our control group is our kind of baseline, and we compare those two groups. If there's a difference between the experimental group and the control group, that's when we say, aha, we have something here, especially if it meets significance, statistical significance, which we'll take a look at in a minute. Remember, our dependent variable in this case is going to be a reduction in like a blood marker for a disease. So we're giving them this shot to see if it helps improve a particular disease. So that's what we're measuring. What happens sometimes is we have our people who are getting the drug, and maybe this drug is helping them. And then we also have people who are in the control group and they're just getting the saline shot. And lo and behold, it seems that they're getting helped as well. The placebo effect is when we see changes in behavior or changes in physiology, even when they are not receiving the drug. So in this particular case, the sample study that I'm talking about here, we're measuring reductions in blood markers for a particular disease. Well, some people, a small percentage of people in that control group might actually show a reduction in blood markers of the disease and report that they are doing much better, and that can be a placebo effect. The placebo effect is when you believe that something's working. When you believe that something's working, this can actually have an effect on your entire physiology. However, it will not be a long-lasting effect. So what will happen is this might improve for a little bit and then go back to their baseline, whatever they started with. So a placebo effect is when you believe that that you're getting the drug and that it's working, even though you're getting the saline. So that's an example of the placebo effect. And here's the definition. Placebo effect, people's expectations or beliefs influencing or determining their experience in a given situation. When people are given a pill to improve their mood may increase just because they believe it will. To know if a medication is really having the effect or whether it was a placebo effect, the experimental group received the medication, control group received a placebo treatment. This is usually done as a double-blind study because we don't want our research who are interviewing these people to be influenced either. Any differences between the groups will be due to the medication. Other issues to consider is the manipulation of variables. As stated, random assignment is required to state causation. We have to make sure that we don't have all of one type of person or whatever in one group and all of another type of person in another group. It has to be random so that your initial population you're looking at, everybody in that initial population or that initial sample of the population has an equal chance of ending up in the drug group or the control group. Sometimes things are not quite that simple. So some experimental designs are more complicated. For example, the effect of sex. When you're looking at differences in sex between males and females on spatial memory, for example. In this case, sex is in a sense our independent variable. In other words, what we're interested in is the differences in sex, but that can't be manipulated. Your subjects are either male or female. So you have to put the males in the male group and the females in the female group. So you cannot randomly assign them in this way. So this kind of experiment is therefore called quasi-experimental. And we're not going to get much into that. I just wanted you to be aware of it. A cause and effect relationship cannot be determined by this type of experiment. All right, moving on to ethics and other issues in research. Some questions cannot be answered using experimental design. And the example of the effect of experiencing abuse as a child on levels of self-esteem. You cannot randomly assign participants to receive abuse. This would need to be studied using another approach, such as case studies or surveys, or a correlational model, where you could look at certain behaviors that people might have, or you can look at levels of self-esteem and see if it's correlated to child abuse or to the type of abuse they received. You could do that kind of study, but we cannot just randomly assign people like, okay, we're going to abuse this child, and then we're not going to abuse these childs, and then we're children, and we're just going to check and see how it goes. Nah, we can't do that. All right, interpreting your experimental findings. Once the data has been collected, a statistical analysis is conducted. We talked earlier saying that you have to use the appropriate statistics. That's important, and knowing how to do the proper research design and using the proper statistics. So that's super important. The statistical analysis determines how likely any differences that you see between your experimental group and your control group are due to chance. Are these just, is this just a minor difference that could be a roll of the dice chance, or is it due to your experimental treatment? Psychologists usually discuss results as significant or non-significant. If the odds that the differences occurred by chance are 5% or less, then the results are significant. There are other experiments that use a different, a different percentage, like that is tighter than that 5%. But for most experimental research, 5% is the level that brings you to statistical significance. A true experiment includes a random assignment, manipulation of the independent variable, and control of extraneous variables that reduces the odds of results occurring by chance. So if you control for all these things that we've talked about that can cause problems in your study, you're more likely, if you see a difference in between your groups, between your experimental and control group, most likely that difference is due to your experimental manipulation And if it reaches that 5% or less, then the results are significant. I know that's confusing, but we're not going to get into statistics in this class. I just wanted to throw that in there. So reporting your findings. As we talked about in our video about doing the paper that you guys have to do, or the papers you have to do, we talked quite a bit about the scientific journals. Once you conduct your experiment and you have got everything done, you've written it all up, what happens then is you submit this to a scientific journal of some sort. And these would be those peer-reviewed journals that we talked about. The aim of these particular journals is to relay your findings to other professionals or scholars in the field. Articles that are published are peer-reviewed journal articles. And once again, the peer-reviewed journal article is an article read by several other scientists, usually anonymously. And the anonymous part is kind of important because you don't want to know whose transcript you're reviewing, and you don't want to know who's reviewing your transcripts. If you know that information, that gives a little bit of a chance for people to do sneaky stuff. So we don't want that. So scientists with an expertise in the subject matter who provide feedback regarding the quality of the manuscript before it is accepted for publication. It helps to weed out poorly conceived or executed studies. It improves articles with suggested revisions, and it determines whether the research is described clearly enough to be replicated by other researchers. So we talked a great deal about replication. Studies have to be able to be replicated. You have to be able to, if we just find that the research, say the researchers did the experiment and they found that this drug works, okay, or this supplement works, and they get it published, and all that's good. But then 3 other people or 4 other people do the same study and they get a different result and say it doesn't work. Well, that's important. At that point, that article maybe needs to be removed, and because there might have been an error in that person's structure of their experiment. So that's why it's important that these are reviewed. So what happens oftentimes is you'll submit the article to the journal, and they'll say, you know what, there is a problem here with this research. You need to compare these other groups or do these other statistics or something like that. So you go back, you do the things they want you to do, you report on it, you send it back in, and then they'll be like, okay, well, this looks pretty good, except you might need some more explanation here. So then you take it back, you correct it again, and then you submit it again. And then finally, after all the things that are unclear or maybe errors or anything like that are taken care of, then they will accept that journal article and then it's published. So it's a really big process. And then once again, replication, I have it on the bottom of the screen here again, because it is super important. It determines the reliability of the original research by being able to have it supported by others. And we've talked a lot about that throughout our other videos. Can include additional measures that expand on the original findings. So you may want to maybe look at a different group using that study, or a different population or something like that, where you can expand that body of knowledge and provide more evidence to support the original finding or cast doubt on those findings. So I can't express enough how important replication is. Bad science and retraction. We've probably all heard the vaccine autism myth, and it is a myth. It is not true. Many peer-reviewed publications published research making claims that routine childhood vaccinations cause some children to develop autism. Well, these reports were kind of large-scale research carried out that suggested that, well, okay, the original, let me just talk without looking at the screen here. Basically, one guy started this whole thing. He did some research and he did not do good research. And he basically manipulated his findings to show that the vaccination was causing autism. And he manipulated these findings because he had a financial interest in establishing a link between childhood vaccines and autism. So he did this for money. So he managed to get this published, and then people started to try to replicate this study to see if it was true, and they found out that it was not true. So this was not something that could be replicated. It was not something that was done correctly or ethically. So this, all of his papers regarding this were rescinded. They were retracted. So the journals sent out, information saying that this article has been retracted, and they basically debunked it and said, okay, this is not true. However, as you're going to learn in our social psychology section, once bad information is put out there, it is hard to undo. And myths like this can run wild. And we see this in a lot of different circumstances. And you guys will learn a lot about this in our social psychology section, because that bad information, once people believe it, it's hard to convince them otherwise. So that's what I have to say about that. Bad research harming a lot of people. Reliability and validity, we've talked a little bit about this, but I wanted to stress this just a little bit more. Reliability is the consistency and reproducibility of a given result. Would the same test give the same result every time? Do the instruments or tools used to collect the data do so in a consistent and reproducible way? So the example I'm going to give you is like a ruler. Say I was using a ruler to measure things. That's my measurement I'm going to use is a ruler. When I have my ruler, which I don't have my prop of a ruler here, but I have a ruler, it's, broken into little sections that are inches or centimeters, and it is reliable. Every time I measure with that, it's going to give me the same measurement. Like I can use this and it's going to tell me accurately how many inches or how many centimeters every single time. The centimeter is the same size every time. The inch is the same size every time. So it is a reliable tool I would use to measure length. So when a study involves observations by multiple people, it is important that they all make observations and record them in the same way. So that's a reliability issue that we talked about when we talked about observational research, that your people need to be very well trained so that they are recording their observations in the same way. Your behaviors have to be clearly defined, all of that we talked about in our observational research section. Inter-rater reliability, that's the measure of agreement among observers on how they record and classify a particular event. That just has to do with the same thing. That's just another term for that type of reliability. A reliable, consistent measurement does not always mean that it is measuring something correctly. So you may have a tool that is reliable. However, it may not be a valid tool for what you're trying to measure. So validity is the accuracy of a given result in measuring what it is designed to measure. Does a test measure what it is meant to measure? A valid measure is always reliable, but a reliable measure is not always valid. Once again, I'm going to use a ruler for this example. Now we have determined that that ruler is very reliable for measuring, for example, length. I can measure how long something is with my ruler. However, if I want to know how much water is in a cup and I put my ruler in there, well, it's going to measure in inches maybe how tall the water is, but it is not a valid tool to use to measure how much water is in that container. It is not a valid tool for that. It's reliable because it will give you inches, but it's not the correct tool. It's not going to measure what you need it to measure. So that's reliability and validity, and those are very important concepts in research. Gotta get a quick drink here. Moving on now to ethics. As we go through our course and you learn some of the experiments that have been done, I think in our learning chapter and our social psychology chapter, that's where we're going to see some big studies that are very important. However, they were done in very unethical ways. And since those studies, ethical bodies that review the ethics of studies that you do have been put in place. So research involving human participants must adhere to strict guidelines. You have to get your research, any of the research you've done has to go, even with animals, has to go through the Institutional Review Board. That's a committee of administrators, scientists, and community members that review proposals for research involving human participants. I don't think we have to have the community for the animal research. It exists at any research institution that receives federal support for research involving human participants. So any of the colleges you go to has an IRB. Generally, IRB must approve research proposals before it can proceed. You have to get that approval. When you are dealing with human beings, it is important that you get informed consent. Informed consent is the process of informing a research participant about what to expect during an experiment and then obtaining the person's consent to participate. This is super important because some of the earlier studies did not do informed consent. So that means that they have to be told what the potential risks are to doing the study. They have to know the implications of the research. They have to be notified that their participation is voluntary. In other words, they can walk out and quit anytime they want. Notification that any data collected will be kept confidential. Their names, any information personal about them will not be reported. So their demographic would, like whether they're male and female, age, that kind of thing, but not, you let them know exactly what you're going to use and what you're not going to use. This is important because, as you will see later when we talk about studies, sometimes in these studies, there have been real harm done to people, and they have been led to believe that they can't leave the study or they can't quit, and it has caused serious psychological damage. A study we're going to talk about in a minute has caused, serious damage and even death to people. And the subjects were not told the potential risks involved. All right, so deception. As I mentioned, in psychology, we often use deception because if people know exactly what you're looking at, it's going to change their behavior. So sometimes deception is necessary to prevent the participant's knowledge of the research question affecting the results as long as it is not considered harmful. So we have to be very careful that the type of deception we use does not cause harm to the person. How do we define deception? A purposely misleading experimental participants in order to maintain the integrity of this experiment. So the example I gave was telling somebody that they were there to for a study involving perception, like sensation and perception, and when it was actually a social psychology experiment. So what you have to do in that case is you have to do what's called debriefing. So in the beginning of the experiment, we tell the person, we do that informed consent, and we're going to tell them, hey, you're actually participating in a study about sensation and perception. That's where we're deceiving them. After the study, we have to debrief them, and we say, hey, by the way, this study was actually about social pressure and social interaction, where we were looking to see if a person's answer could be influenced based on social pressures in the room or something like that. I'd have to tell them exactly what it was about, and then answer any questions that they have, and all of that. So there's a lot to that. So one of the most horrific, well, there's a lot of horrific studies that we have done in the past, a lot of them involving Black Americans. There's so many with like mustard gas and all sorts of things that they were, that the Black community was subject to. We're going to give a quick example that your book gives, and that is the Tuskegee syphilis study. So ethical guidelines have not always existed, as we talked about. So in 1932, there was a total of 600 participants in the study that was studying syphilis. Now in 1932, there was no cure for syphilis. We didn't have like penicillin to give them, anything like that. So they were recruited. Participants that tested positive were not informed that they had the disease. So they tested 600 people as Black men, 600 Black men. They were not told whether or not they had syphilis. What they did was 400 had syphilis, 200 did not. And then they just kept watching the progression of the disease in these people. They just monitored and watched them progress. And syphilis is quite horrible. It causes severe damage to the brain. So towards the end of life, you have some super bad mental health issues. No cure existed at the beginning of this study. However, in 1947, a cure was found. And the cure was found in 47, which is penicillin, but guess what? These people were not administered penicillin. They just watched them die. So this study went on, and I unfortunately, I don't have my notes here. I don't know what year the study ended. I think it was in the 50s that it ended. And at the end, there was only like a couple hundred people left alive in the study. And they could have saved these people's lives by giving them the penicillin. These people also ended up spreading the disease to family and other people because of their lack of knowledge that they even had syphilis. And I don't know what the researchers told them, but they deceived them, and these people died horrific deaths because of the deception and the lack of treatment they were given, the unethical treatment. And we're going to see a lot of things like that we talk about that in the history of research. Not just, this one is more of a medical research thing, but we're going to see things that are very sketchy that we see in psychological research as well. So this is why the ethical, this is why we have these panels that take a look at these research studies to see if they're ethical and if you could go ahead with them. All right, ethics involving animals. Basically, the Institutional Animal Care and Use Committee. This is another committee that you have there at the school that's going to help you help you determine whether or not your study is ethical. They're not going to let you just do research on an animal for no reason. 90% of psychological research involves animal subjects using rodents or birds, because we're learning a lot about brain activity, brain stuff, behavior, and using animals is a pretty good model. All right, so animals used in research. We do that often when it's unethical to do in human participants. Now, I used to do a lot of animal research. I don't do it now. I actually became, the rats won. I got really allergic to them. They almost killed me. So they got the last laugh at me. I had to do some really serious soul searching before I participated in animal research, because I had to, even though the study that I wanted to do was approved, I had to make sure that I felt that it was important enough of a study to have to put these animals through what they went through, which was just basically a learning study, but then I took their lives at the end and studied their brains. So I had to make sure that this was something that I felt was important enough, and of course that the committee would also approve. So using these animals, you have to reduce the pain and distress of the animals. You have to do things in the most humane way possible. And rats are adorable, by the way, just in case you didn't know, they're super cute and adorable. All right, that is it for this video. I hope you find it useful. I am doing this in my living room on the weekend, so there's people wandering in and out, so I get a little distracted and I get a little kind of off track occasionally, and I apologize for that. I'm doing the best I can, but this is how it has to be. So you guys take care, and I look forward to our next video.